Okay, so let's talk about Chapter 6, cells. Um, chapter starts with a graphic here that just shows the size of various things and the degree to which we can see these things using different method. Of course, with our eyes we can see quite large things, and there are some larger cells that is possible to sort of see with your eye. Some neurons are quite long, although you don't really see the individual neuron. You can see clusters of them, and some eggs are kind of large. But to see cells for the most part, you need microscopes. Um, Light microscopes that we use, of course, are very good for seeing cells and sometimes individual components of cells. Now, to see some of the really small stuff, though, you need an electron microscope. Um, there is a detailed discussion of microscopy in the chapter and different techniques. You don't need to worry about knowing that. You should be familiar with this, though. It's a technique called cell fractionation, where you can basically take cells and divide it up into different fractions or parts, if you will. Um, First, by grinding up the cells using a high-tech piece of machinery called a blender. Um, Then once you've done that, you've busted up the cells, you start to centrifuge it at different speeds for different times. So you do it at a relatively slow speed at first, and that gets rid of relatively large materials. Then you take that liquid off, spin it again at a higher speed, and you can get smaller objects like the mitochondria, chloroplasts. Then you can take that liquid and spin it at an even higher speed, and you can start to get even smaller parts, pieces of membrane, for example. Then you can take the remaining liquid and spin it at an extremely high speed and get the smallest things such as ribosomes. Um, so it's just a way of using grinding and centrifuging to separate the parts of the cell. All right, so the simplest cells out there, I guess we could say, are bacterial cells. Well, of course, what we know as prokaryotes. Okay. Now they have everything a cell needs, but are lacking things that eukaryotes have. Of course, they have a cell membrane like all cells. They have some DNA. It's not contained within a nucleus though, and they don't have. We would say prokaryotes are lacking membrane-bound organelle. Membrane-bound organelle. So they don't have things like mitochondria and chloroplast and endoplasmic reticulum and Golgi, all that good stuff. They do have ribosomes, though, because they do have to make proteins. The ribosomes are just within the cytoplasm. The DNA is sort of clustered in a region we call the nucleoid. Of course, we have a cell or plasma membrane. Bacteria typically have a cell wall outside the cell membrane, helps them maintain shape. They'll often have a capsule outside of that, a material they secrete that um, can serve a protective function and also help hold them in place. Some bacteria do have flagella. We'll see that the structure of flagella in bacteria is different from the structure of flagella in eukaryotes. So they have the same function of movement, but the fact that they have a different structure suggests to us that they evolved separately. Um, you can see the approximate size. That's a half of a micron. You have 1,000 microns in 1 millimeter. So this is pretty small size. This guy's about a half micron wide and maybe, I don't know, 2.5 microns long. Um, Bacteria generally are less than 10 microns in size. All right. Um, so we'll get to eukaryotes in a second, but just a brief discussion about surface area and surface to volume ratio. Um, so this idea of the surface to volume ratio plays an important role with cells, and it's why cells generally tend to be small because, as is shown with these cubes here, of course these are the same shape, but clearly this one is bigger. And so the surface size, of course, is much bigger, and the volume is much bigger as well. But what you notice is that as you go from a smaller to a larger, similar sized object, the volume increases at a greater rate than the surface area. Thus, your surface to volume ratio decreases because, again, the volume has increased more, and that's because surface area is a two-dimensional characteristic, and volume is a three-dimensional characteristic. And so, while surface area is increasing in those two dimensions, volume is increasing in three dimensions, so it increases at a proportionally greater rate. I like to think of it this way as your volume is increasing at this rate, but your surface area is increasing at this rate, but your volume is increasing at a greater rate, such that, again, surface to volume ratio decreases as something gets bigger. So what does that have to do with cells? Well, I don't know if you just think of a cell as being kind of roundish. Cells have to get things in and out of them. And when you think if you're in the center of a cell, more or less, or somewhere deep inside the cell, when the cell is larger, you are further from the edge, further from the cell membrane. And that's a greater distance over which things have to move. And so with greater size, it is more difficult for cells to get things in that they need and get rid of waste. And so the cell has to basically work harder to do that. And so it's more efficient in terms of getting things in and out for a cell to be smaller because there's more surface area relative to the volume. All right, so animal and plant cells, your typical eukaryotes, although eukaryotes also include protists and fungi, as we'll see, but we'll focus on plant and animal cells here. All sorts of extra interesting parts now, all these internal membranes, the endoplasmic reticulum, well, first, of course, the nucleus with its membrane, the endoplasmic reticulum, Golgi, mitochondria, etc. We'll talk about these individually. What we want to focus on here, though, is just some of the differences between plant and animal cells. Animal cells have a more extensive cytoskeleton, as we'll see. They have, whoops, whoa, go away. They have this thing called the centrosome that you don't see in plants. It's involved with producing microtubules and cell division. And they have lysosomes that you don't see in plants. These are organelles that contain digestive enzymes. But everything else, they typically, this shows not having flagella in plant cells, but you do have some in a couple groups of plants, in particular mosses and ferns, they do have flagellated sperm cells, just like in animals, but in flowering plants and conifers, you don't. So I guess you could say their presence is more limited in plant cells. Okay, plant cells. Again, a lot of the same things, mitochondria and Golgi and ER, nucleus, all that. But what do they have that animals don't have? The central vacuole, this large, mostly water-filled sac inside the cell that helps maintain the water pressure inside the cell. Cell wall, you don't see that in animals, gives it support. And so because they have a cell wall, they have a less extensive cytoskeleton than animals. Let's see, plasmodesmata. These are cytoplasmic connections between adjacent cells. Animal cells, as we see, have connections. They sometimes have connections between cells, but they're not known as plasmodesmata, and they're somewhat different. Let's see. Okay, So I guess that's some of the basic differences between plant and animal cells. Let's do section 3. The nucleus. So starting inside of our typical eukaryotic cell, we now have the DNA housed within the structure we call the nucleus. Um, in your typical undividing or not getting ready to divide cell, the DNA is in a relatively diffuse state we know as chromatin. It's just kind of spread throughout the nucleus. And if you stain that cell, the whole nucleus shows up as this dark mass of DNA. Um, the individual chromosomes are there, it's just that you really can't see them because they're all spread out and sort of intertwined. Nucleolus is a region in the nucleus where RNAs and ribosomes are being made. Okay. Those materials will be exported from the nucleus to be used outside in the cytoplasm. Um, nuclear envelope has a double membrane. It's made of phospholipids, just like any cellular membrane. There are these openings, these pores, that are formed by proteins that um, allow for movement of things in and out of the nucleus, primarily uh, a way for RNAs and ribosomes to get out of the nucleus. All right, you see there's endoplasmic reticulum, so there's a direct connection um, to the nuclear envelope of the endoplasmic reticulum, although there are sometimes endoplasmic reticulum that's not connected to the nucleus, but it often is. Rough ER means it has ribosomes attached, and because of that, you can suspect you're going to be making proteins at this rough ER. That's the primary job of the rough ER. We'll see there's also smooth ER. Um, so when DNA, so the blue ribbon here represents the DNA, and you can see it's wrapped around these proteins. These proteins are known as histones. They come in clusters of 8 that they wrap around. So when the DNA is sort of in this state, that's what we refer to as the chromatin state. It's spread out. It's still attached to the proteins, but spread out. But when it starts to condense, that's when a cell is getting ready to divide, and it's the DNA is condensing up. You can begin to see distinct chromosomes. As we'll see, when those chromosomes condense up, it makes it a lot easier for cell division to happen. Um, so ribosomes attached to endoplasmic reticulum, but you also have free ribosomes out in the cytoplasm, or in the cytosol, the liquid part of the cytoplasm. Ribosomes, the sites of protein synthesis, they consist of both ribosome RNA and protein, and they come in what's called the large subunit and the small subunit, and they get together to make the ribosome. Um, all right, I'm just about out of time, so we'll go on to the other sections in another video.